What is the key difference between embryonic and iPS cells? Imagine being able to create versatile cells that can turn into any part of the body. That's what stem cells do, but not all stem cells are the same. Today, we're exploring the main difference between embryonic stem cells and induced pluripotent stem cells. Embryonic stem cells come from very early-stage embryos. These are tiny clusters of cells from a fertilized egg that has not yet developed into a baby. To get these cells, scientists usually take a small part of an embryo that is about 5 to 6 days old. This process involves destroying the embryo, which raises ethical questions for many people. Because they come directly from embryos, embryonic stem cells are considered the most flexible and are often called the gold standard for pluripotency. On the other hand, induced pluripotent stem cells are made by reprogramming adult cells. Usually, scientists take skin cells or blood cells from a person and introduce special proteins called transcription factors. These proteins tell the adult cells to revert back to a flexible, stem cell-like state. This process can take a few weeks and involves changing the cell's gene activity. The result is a cell that can become any type of tissue, just like embryonic stem cells. Both types of cells can develop into any of the 3 main tissue layers in the body, which makes them very useful for medical research and potential treatments. They can also renew themselves indefinitely under the right conditions. But the key difference is where they come from. Embryonic stem cells are naturally derived from early embryos, while induced pluripotent stem cells are created in the lab from adult cells. This difference is important in ethics. Using embryonic stem cells involves destroying an embryo, which some believe is morally wrong because it ends potential human life. Many countries have strict rules or bans on embryonic stem cell research for this reason. Conversely, induced pluripotent stem cells do not requiring embryos. They are made from adult cells, which many see as a more acceptable option ethically. Scientifically, there are some subtle differences. Induced pluripotent stem cells may carry epigenetic marks from their original adult cell type, which can affect how well they turn into different tissues. Also, reprogramming can sometimes cause genetic mutations or abnormalities. Embryonic stem cells tend to be more uniform because they come directly from early embryos. In practical use, induced pluripotent stem cells are promising because they can be made from a patient's own cells. This reduces the risk of immune rejection and sidesteps many ethical issues. However, embryonic stem cells are still very valuable for understanding early human development and serve as a benchmark for testing the quality of induced pluripotent stem cells. So, the main difference is that embryonic stem cells come from early-stage embryos, raising ethical concerns about embryo destruction. Induced pluripotent stem cells are created by reprogramming adult cells, avoiding those ethical issues but with some biological differences that are still being studied. Both types hold great promise for future medical advances and continue to shape the way we think about ethics in advanced medical technologies.